CRISPR-Cas9 has taken the world by storm with the promise of making gene editing much easier and faster than ever before. But how does CRISPR actually work? How can biology research benefit from it? What will happen when we start using it to edit human DNA? And what's the fight between its developers all about? CRISPR-Cas9 is one of the biggest discoveries of the 21st century. Since it was developed in 2012, this gene editing tool has revolutionized biology research, making it easier to study disease and faster to discover drugs. The technology is also significantly impacting the development of crops, foods, and industrial fermentation processes. But the one application that has made it famous is the modification of the human genome, which brings the promise of using CRISPR to cure disease. The first clinical trials testing CRISPR-Cas9 in people are already underway in China, Europe, and the US. So while scientists start venturing into tweaking our own DNA, it is worth taking the time to fully understand what CRISPR is, and what the actual benefits and risks of using the technology are. First of all, what is CRISPR-Cas9? CRISPR technology is a simple yet powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. Its many potential applications include correcting genetic defects, treating and preventing the spread of diseases, and improving crops. However, its promise also raises ethical concerns. In popular usage, CRISPR is shorthand for CRISPR-Cas9. CRISPRs are specialized stretches of DNA. The protein Cas9 is an enzyme that acts like a pair of molecular scissors, capable of cutting strands of DNA. CRISPR technology was adapted from the natural defense mechanisms of bacteria and archaea. These organisms use CRISPR-derived RNA and various Ca proteins, including Cas9, to foil attacks by viruses and other foreign bodies. They do so primarily by chopping up and destroying the DNA of a foreign invader. When these components are transferred into other, more complex, organisms, it allows for the manipulation of genes or editing. What can CRISPR do? In theory, CRISPR gene editing could be used to make any modification to the DNA of virtually any living being. In biotech and pharma companies, CRISPR is becoming the go-to tool for drug discovery. In academic research labs, the gene editing tool is being used to modify the genome of all sorts of organisms to study the function of any gene of interest, whether it is one that causes disease or one that makes a crop grow faster or survive harsh conditions. In agriculture, CRISPR could be used to produce crops with better yields or that resist drought, much faster than is possible with traditional breeding techniques. It can also be used to add new features, such as making tomatoes spicy, or remove others, for example making gluten-free wheat or decaf coffee beans. However, regulations can limit the use of these technologies. While the US has already seen the launch of CRISPR-modified crops, the European Union decided to set strict GMO regulations that scientists believe are hindering the potential of the technology. But right now, most of the money seems to be in using CRISPR-Cas9 to engineer human DNA. With over 10,000 diseases caused by mutations in a single human gene, CRISPR offers hope to cure all of them by repairing any genetic error behind them. There are two main approaches to using CRISPR as a therapy. The first is called ex vivo gene editing. It involves extracting human cells, engineering them in the lab, and re-injecting them into the patient. This method is similar to that used for most gene therapies already on the market. However, it can become quite expensive given each patient requires an individual manufacturing process for their therapy. The second method is called in vivo gene editing and involves delivering CRISPR-Cas9 into the patient's body to edit the DNA directly from within the cells. CRISPR could be delivered inside nanoparticles or encoded into DNA and be cleared out of the body once it has completed its mission. This method has only started human testing in 2020, and there are some concerns that there is a risk of CRISPR making off-target modifications. Wait, is editing human DNA with CRISPR safe? Or ethical? Those are the big questions right now. especially after CRISPR gene editing was controversially used to create the world's first gene-edited babies in 2018. These CRISPR babies carry a mutation intended to protect them against HIV infection. The experiment resulted in a strong pushback as scientists around the world questioned the ethics of altering human DNA without fully understanding the possible consequences. Indeed, there was a study suggesting that people carrying these mutations might be at risk of catching certain infections and dying younger. Despite all the controversy around CRISPR therapy and the large amounts of money invested in it, we are still in the very early stages of clinical trials. Scientists are wary of repeating the same mistakes as when gene therapy was first tested in humans back in the 90s, resulting in the death of 18-year-old Jesse Gelsinger and causing years of delay in the development of gene therapy. Now, even if CRISPR proves to be safe in humans, is it ethical to modify the human genome? The first applications of the technology, aimed at curing genetic diseases, seem quite straightforward. But where should the line be drawn? At what point does a therapy become a tool for eugenics? Although the point in time when we are able to modify all sorts of human features at will is far ahead in the future, it is never too early to start thinking about how the technology should be regulated. Many scientists, including Jennifer Doudna, seem to agree that we should stay away from germline editing, that is, any modifications that children will inherit. At least for now. Who is developing CRISPR-Cas9 therapies? Since the first publications showcasing CRISPR-Cas9 as a gene editing tool back in 2012, a number of companies have been set up by the developers of the technology. Based in Switzerland and the US, there is CRISPR Therapeutics, co-founded by Emmanuel Charpentier. Working in partnership with Vertex Pharmaceuticals, the company already has preliminary results showing promise for the treatment of the blood disorders beta thalassemia and sickle cell disease. The company is using an ex vivo approach, where the bone marrow stem cells of the patient are genetically engineered outside the body. In the US, the first CRISPR clinical trial started in early 2019, run by scientists at the University of Pennsylvania. On top of academic efforts, the US counts also with the firm Intellia Therapeutics, co-founded by Jennifer Doudna. Its first target will be an in vivo treatment for a rare neurological disease called transthyretin amyloidosis. Founded by Doudna and Charpentier's competitor Feng Zhang, there is also Adidas Medicine, working in therapies for genetic blindness and cancer, among others. Doudna originally co-founded Adidas along with Zhang, but stopped all involvement with it just a few weeks after Zhang was granted his CRISPR patent and issues concerning intellectual property began to appear. What's next for CRISPR? With its potential already demonstrated in research applications, the next big milestone for CRISPR will be proving to be safe and effective as a treatment. But there are still many other applications underway. For example, the US company eGenesis, co-founded by George Church, is using CRISPR to modify the pig genome so that their organs can be transplanted into humans without rejection. Another is the use of CRISPR as a diagnostics tool. Some researchers have already developed a method to detect COVID-19 using CRISPR. CRISPR might still surprise us as new variants are developed. Swiss scientists have developed a method to simultaneously edit up to 25 genes using CRISPR. And another version of the gene editing tool called CRISPR-CPF1 that makes it easier to replace one DNA sequence by another is already being used by big brands such as BASF. You can imagine that many labs including our own are busily looking at other variants and how they work, Doudna said. So stay tuned. The gene editing tool is also becoming very popular between DIY scientists and biohackers. Some believe that the relatively simple methods that this technique requires might help democratize science and bring it closer to people outside the lab. However, some cases of biohackers injecting themselves with experimental treatments have alarmed the public, and it remains to be seen how these uses will be regulated. In any case, the impact of CRISPR in biology is already tangible and will undoubtedly go down in history as a big discovery. The cherry on the cake will come when the technology wins the Nobel Prize, which many have been unsuccessfully predicting will go to CRISPR for years. It is possible they are waiting for CRISPR to demonstrate all the potential that is expected, but it would be unfair, CRISPR discoverer Francis Mojica told me. What CRISPR has already achieved is much more than what other tools that have received the Nobel have achieved. The prize has gone to tools used to cut and copy DNA in the test tube. CRISPR can be used to edit genomes, change expression levels, visualize DNA, kill bacteria, develop diagnostics, and many more applications, even to store a movie within DNA. I am convinced it will get it. When? I don't know. Thank you for watching the video. See you in the next science video. Don't forget to like the video and write your thoughts in the comments section. I'd appreciate it if you'd subscribe to my channel.